Hello, my name is Brittany Hancock-Brown, and I'm here to tell you about a genetic disorder called myotonic dystrophy type 1, or DM1 for short. DM1 is a genetic neuromuscular disease that globally affects 1 in 8,000 people and has different distributions in different countries. Let's go over some genetic basics. Humans have 23 chromosomes, which contain around 19,000 protein-coding genes. These genes make up nearly every part of who we are, and small changes to certain genes can have big impacts. For our discussion of DM1, it is also important to note that genes include components called introns and exons. When the DNA is read to make RNA, which will be used to code for proteins, the introns are cut out and only the exons are used. In the case of DM1, the defect involved is found on the 19th chromosome on the gene called DMPK. DMPK has 15 exons, and the defect is found on the 15th exon in the sequence. Our DNA is made up of nucleotides A, T, C, G, and arranged into units of 3 called codons. On exon 15 of the DMPK gene, there are repeats of a trinucleotide sequence, CTG. The average person has 5 to 34 copies of the CTG repeats, but those with DM1 have an excessive number of copies. The most mild form of DM1 has 50 copies, with the most severe cases having over 4,000 copies. Let's talk about the symptoms and severity of DM1. There are 2 types of DM1, adult onset DM1 and the more severe congenital DM1. DM1 typically affects multi-system function. In the cardiovascular system, DM1 can cause atrial arrhythmias and heart blockages. In the digestive system, it can cause formation of gallstones, constipation, and difficulty swallowing. In the reproductive system, it can cause hypogonadism and testicular atrophy. In the muscular system, it causes myotonia, or the inability to relax muscles, as well as muscle weakness and muscle wasting. In the nervous system, it can cause some cognitive deterioration, speech disability, daytime sleepiness, and cerebral atrophy. The most common symptom of DM1 is the formation of cataracts. In congenital DM1, the symptoms include poor feeding capability, respiratory distress, facial weakness, and mental retardation. When discussing a genetic disorder, it is always appropriate to address inheritance. DM1 has what is called autosomal dominant inheritance. This type of inheritance means that if you have one parent affected with the disease, then there is a 50% chance that the child will also inherit the disease. However, it is worth noting that almost all of known congenital cases of DM1 are a result of the affected parent being the mother. Another interesting feature of DM1 inheritance is a phenomenon called anticipation, whereby successive generations of offspring with DM1 will have a more severe case of DM1 than the previous generation. In DM1's case, it means that the longer the lineage of the persons affected with DM1, the more CTG repeats present within the individual's DNA. In rare cases, about 5%, reverse anticipation occurs, and the number of repeats decreases. Now that we've reviewed the basics of DM1, let's talk about the mechanisms. The question here is, why are CTG repeats detrimental, and how do they cause the symptoms presented in DM1? DMPK is the gene where the mutation that causes DM1 occurs. It is a gene that codes for the protein myotonin protein kinase. This is a distribution chart of where myotonin protein kinase is concentrated. Recognize any similarities between these regions and the symptoms listed earlier? Actually, even though DMPK is the gene with the defect, the protein it codes for is hardly affected. In fact, we are generally unsure of what the function of myotonin protein kinase is. So how does the expansion affect those with DM1? Along the way to becoming a protein, a process called transcription occurs, where the DNA is turned into mRNA, where it will eventually be translated and used to instruct protein synthesis. The mRNA produced by the mutant DMPK contains CUG triplet repeats. These CUG repeats attract and gather a different protein, MBNL1, rendering MBNL1 functionless. This is important because MBNL1 functions as a regulator in an important genetic process called alternative splicing. While there are around 20,000 protein-coding genes in the body, some genes are capable of coding for more than one type of protein. They do this by alternative splicing. When certain conditions are present, exons can be included or excluded from mRNA. The mRNA is what will eventually be used as instructions for building the protein. Depending on which exons are included, different proteins can be made. In those affected with DM1, alternative splicing is misregulated and is thought to cause up to 24 different misregulated splicing events, which has numerous effects. For example, myotonia, the inability to relax muscle, is caused by the misregulation of splicing in skeletal muscle CLC1 or chloride channel 1 via the inclusion of exons with premature stop codons, which downregulates CLC1. Some other examples are increased inclusion of CTNT exon 5, which causes cardiac conduction defects, and skipping the insulin receptor on exon 11, causing increased insulin resistance. Do you have a better understanding of what DM1 is? Did you find it interesting to learn about a genetic disease that affects over 900,000 people worldwide? Online resources in the field of genomics makes information like what I've presented today widely available to anyone who knows where to look. Genomics is the study of the function of genes and the genome as a whole, as well as related techniques. Here is a list of some resources that can help you get started researching a genetic disease, such as DM1. First, a simple Google search can help you find a disease of interest. You can look up genetic diseases and navigate to a website with a list, such as medicinenet.com. Once you've found your disorder of interest, the website omim.org is a good place to start when looking for in-depth information. On the disorder page, you can find the gene that your disorder stems from. OMIM provides lots of helpful information about the disorder and can direct you to research articles. Now that you know the gene responsible for your disorder, you can use that in searches on other genomics-based websites. Ensemble.org is a good website to get detailed information about your gene and to take a closer look. Here we can look at the region in detail and have a good look at DMPK and the details surrounding the gene. Another interesting website is Expression Atlas. Here you can look at the differential expression of your gene of interest in many different conditions and different diseases. There are many other resources available for genomics that can help anyone gain a better understanding of a disease and can help researchers lay the foundation for future experiments and perhaps even come up with more conclusions based on existing data. I hope that you enjoyed this video and that you learned something new. Thanks for watching.